We have Sarah Moretto from State University Londrina in Brazil, and she will be presenting her analysis of genetic polymorphisms of the PD-1 gene in breast cancer patients. Sarah, do you mind sharing your screen?
Yes, can you hear me?
Yes.
Okay, good. I'm going to share my screen. Can you see my presentation?
Yes.
Yes?
Yeah, you're good to proceed. If you want to share your, turn your camera on as well.
Turn my camera off?
Oh, on.
On? Oh, sorry. Okay. So, good afternoon, everyone. My name is Sarah. I'm from State University of Londrina. And the title of my study is Analysis of Genetic Polymorphisms of PD-1 Gene in Breast Cancer Patients. So, in Brazil, cancer is the second leading cause of death amongst men and women, just after circulatory system diseases. Among women, breast cancer is the main cause of cancer and cause of death. Around 66,000 new cases per year in the triennium of 2020 to 2022, which represents around 30% of all female cancers diagnosed, with the exception of non-melanoma skin cancer. Breast cancer can be histologically classified as lobular and ductal. The ductal carcinomas represent most of the cases, around 80%, and lobular represents 10 to 15% of the cases. We also have the TNM staging, which was established by the Union of International Cancer Control, and TNM stands for tumor, lymph nodes, and metastasis status, and it ranges from 0 to 4, in which 0 is the best outcome stage and 4 is the worst outcome stage. We also have a molecular classification based on a few molecules that we use as markers. A few of these molecules are estrogen and progesterone receptor, HER2 receptor as well, and the marker of proliferation K167. Along all these biological markers, we can divide breast cancer in 4 main subtypes, which are luminal A, luminal B, HER2, and triple negative breast cancer. Triple negative being the worst type of breast cancer with the worst prognosis. We also know that the tumor microenvironment is an intricate relationship between tumor cells and immune cells, and these interactions are complex and may contribute to the interaction, growth, invasiveness, and metastasis of these tumors. Amongst all the immune cells that we can find on the tumor microenvironment, which are just displayed here on your right, I highlight the TCD8 lymphocytes, because the TCD8 lymphocytes have the anti-tumor activity that can protect the host against tumor cells. However, there are molecules that can interfere. These molecules are called immune checkpoints. And one of these immune checkpoints is the PD-1 receptor. PD-1 is the programmed cell death receptor, and when it's connected to its ligand, the PD-L1 that is expressed on the surface of tumor cells, it inhibits the anti-tumor activity of TCD8 lymphocytes, allowing therefore tumor evasion. We also know that many genes in our body have many genetic variations, and PD-1 also has genetic variations. One of these genetic variations we call single nucleotide polymorphism. That is a change in a single nucleotide in a specific region of the gene. PD-1 gene is located on chromosome 2 and has 4 introns and 5 exons. We analyzed in this study 2 polymorphisms located on intron 4. They are very close to each other, and they have the variations guanine to adenine and cytosine to thymine. Therefore, the aim of our study was to analyze genetic polymorphisms of PD-1 receptor and its possible implications in the pathogenesis and prognosis of breast cancer, comprising all the subtypes of this disease, which are luminal A, luminal B, HER2 enriched, and triple negative. This study has been approved by the Ethics Committee of State University of Londrina, and we have gathered 400 control individuals that were neoplasm free, and we obtained peripheral blood samples from these individuals. We also obtained peripheral blood samples from breast cancer patients, 204 breast cancer patients, and also clinical pathological parameters were obtained, and these include tumor size, lymph node commitment, K167 index, histological grade, estrogen and progesterone receptors status, and also HER2 status, and the TNM staging. Following the DNA extraction from the blood samples, we proceeded to PCR reaction to amplify the region of the polymorphisms. In one single PCR, we could amplify the region for both polymorphisms. They are very near to each other. And after confirming the amplification through electrophoresis, we followed with enzymatic digestion to genotype these individuals. Here you can see on this image on your right, a picture of an acrylamide gel with all the profiles from each polymorphism. So our first results regarding the frequencies and the characteristics of our groups were that the breast cancer patients had an age ranging from 25 to 99 years old, and the mean age of these patients was 54 years old. For the control group, the age ranged from 18 to 87 years old, and the mean age was also 54 years old. 95.6% of the tumors were ductal carcinomas, and 2% were lobular carcinomas. The mean tumor size of our patients were 2.9 centimeters. On this table, you can all see the allelic and genotypic frequencies of both polymorphisms from each group, the control, total breast cancer group, and the subtypes of breast cancer. We follow with the statistical analysis, and we found on our case control association study a association between the A allele carriers of the GA polymorphism and the risk of developing breast cancer. However, we didn't find the same results for the CT polymorphism. Regarding the clinical pathological parameters for the GA polymorphism, we also found a positive correlation between estrogen and progesterone status. However, these results were not observed for the CT polymorphism. So we know that regarding the GA polymorphism, the transition between G to A leads to a disruption of the binding site of the RUNX1 transcription factor. And this disruption may lead to alterations of the transcriptional regulation of the expression of PD-1 gene, leading to an alteration of the expression of the receptor on the surface of the T cells. This could lead to an augmented T cell activity. However, the increased binding of PD-1 to its ligand PD-L1 may lead to exhaustion of the T cell. And this exhaustion leads to an inhibition to anti-tumor immunity, therefore allowing tumor evasion to occur. Also, the absence of correlation that we observed between CT polymorphism and the risk to developing breast cancer can be justified by the population studied. We know that the contribution and the influence of polymorphisms to developing diseases can depend on the population and also the pathogenesis and etiology of the disease. Therefore, further investigations on different populations and increased number of populations remain to be done to confirm if there is or not an association between this polymorphism and breast cancer. The correlation that we found between the GA polymorphism and the estrogen receptor status can be justified by a very recent study that confirms that ER status can be an important factor that can affect the levels of PD-1 on breast cancer tissue. Also, the correlation we found between progesterone receptor status can be explained by this study done in 2016, in which they found that hormones such as progesterone can lead to an increased PD-1 expression on many immune cells. Therefore, further investigations regarding the expression of PD-1 on breast cancer tumor cells remain to be done to confirm this correlation. In conclusion, we can say that these findings can contribute to the comprehension of the interaction between PD-1 polymorphisms and breast cancer susceptibility. In addition, the PD-1-GA polymorphism can be considered as a potential marker of susceptibility and prognostic for breast cancer. I'd like to express my thankfulness for the Cancer Hospital of Londrina for the samples, the financial institutions, our laboratory, and all the people in the lab that contributed for this research, and Londrina State University for the great opportunity. I'd like to thank you all for the attention and the opportunity for presenting my work. Thank you very much.
Thank you, Sarah. That was a great presentation. Really interesting topic.
Thank you.
The attendees have a question. Dr. Eleanor, do you have a question?
Sure. I just wanted to ask a quick question. So you've got here a PD-1-GA polymorphism as a good biomarker, and I'm wondering whether in different populations, different ages, you see variations, or how homogeneous or heterogeneous was that hospital sample?
Yeah, it was, I would say for our samples, they were quite homogeneous. But when we compare to studies abroad, for example, studies in Pakistan, studies in China, there are very different frequencies of these, the allelic frequencies and genotypic frequencies. So there are studies, there is one study that studied breast cancer patients and the GA polymorphism, and they did not find any correlation at all with any parameter. This is the first study that finds correlation between this polymorphism and the parameters of breast cancer. So it's still a very scarce answer field. We are looking forward to get more answers in the future. It's very exciting.
Maybe just a quick question. I don't know if we have any. Actually, we do have one from the Q&A. Elliot, why don't we go ahead and ask that one first?
Sure. An attendee is asking, what is the correlation between PD-1 and response to therapy?
That's a great question. We have immunotherapy with antibodies that bind to PD-1 and PD-L1. We still don't know the correlation between these immunotherapy and the actual polymorphisms. PD-1 has many polymorphisms on exons, introns, UTR regions. So it's a variety of variations that remain to be studied yet. We don't know if these polymorphisms have a direct influence on the immunotherapy, but I believe there is, but I don't think it was found yet.
Thank you.
So maybe kind of linking it back to Anthony's presentation a little while ago, what do you think about the potential power of using something like single cell to try to look at a signature of polymorphisms as opposed to one at a time to try to get a better diagnosis, especially for some cancers, let's say pancreatic cancer, we know is much more difficult to treat with immunotherapy. What do you think is the right way for moving forward for getting a better, more complete signature for prognosis?
I think that's very individual. Here it's a very basic science that we do in our lab. It's not really applied on the clinical field, but I believe that understanding these variations can lead to a complete screening of the patient and a very individualized treatment regarding immunotherapy, chemotherapy, or radiotherapy. So I think the expectations are great for that. And recently PD-1 has been growing in attention because of the Nobel Prize, right? and in 2018, if I'm not wrong. So I think that we will find many good things in future, many helpful things.
Thank you. Really great presentation.
Thank you.
Yes, thank you, thank you. Oh, I believe we have another question really quickly. What is the specificity of PD-1 as a diagnostic tool compared to and combined with other markers?
We have established markers for breast cancer, such as the BRCA gene mutations and P53 mutations. So PD-1 is very general regarding all cancer types, not just breast cancer. So the profile of expression of PD-1 on breast cancer is very varies from population to population. That's why it's important to study specific populations. And in Brazil, we are very heterogeneous in population. So that's why we have sometimes crazy results like this, and it's difficult to compare to other studies in other countries. But I think that when we understand exactly the function of the PD-1 on breast cancer and how it interferes, I think it can be very used as a marker on breast cancer.
Awesome. Thank you for sharing. And with that,